Hi, thanks for joining us for today's webinar. I'm Esther Marco, Product Specialist at FEMTONICS, and I also have Dr. Tamás Tompa here with me today, who is an application specialist with 10 plus years of experience in multiphoton microscopy and physiology. Although we are not wearing masks, we did undergo COVID testing today, which came back negative, and we strongly advise everyone to wear masks according to their country's regulations. So, about today's topic, uh, FEMTO3D Atlas is our flagship 3D multiphoton microscopy platform that can capture fast fluorescence changes from network to dendritic spine scale. There is no similar microscopy platform that can fulfill the needs of those asking important questions about the functions of cellular networks in large brain volumes. And today we want to show you what kinds of scientific breakthroughs are possible for those who embark on using the FEMTO3D Atlas technology. And quick information about the flow of the webinar. Please drop your questions into the Q&A option at the bottom of your Zoom application, and we will get back to your questions at the end. In case we run out of time before getting to your question, we will make sure to send you a reply to the email address which you used to register for this webinar. And with that being said, let's start today's presentation.
Enjoy.
FEMTO3D Atlas offers you an all-in-one solution for 2D and 3D imaging. In this overview slide, 5 features are summarized that raise FEMTO3D Atlas well above the level of competing conventional galvo or resonance scanner-based systems. First, let's look at the high-speed arbitrary frame scanning mode. In its basic form, this mode enables you to perform planar frame scanning, known to most of you who have already used a conventional confocal or multiphoton scope. Even in this basic mode, Atlas offers more than the best resonant galvoscopes, both in acquisition speed and in flexibility. The video illustrates FEMTO3D Atlas's advantages. You can see neurons labeled with GCamp recorded at 40 frames per second, faster than resonant galvoscopes. In addition to speed, high-speed arbitrary frame scanning gives you the unique possibility to rotate the field of view to any angle in 360 degrees. This can be very useful when the object of interest is not exactly parallel to the objective lens, which is almost always the case. Second, Atlas can offer 4D volume scanning, a very important capability when you want to capture signals in all 3 dimensions, not just at the optically sectioned single plane. Third, Atlas breaks the limits of frame-based 2D scanning and gives you the option to record from locations scattered in a volume via 3D region scanning. This mode opens an unmatched range of possibilities that are not available in conventional systems. Fourth, the 3D anti-motion technology cancels out motion artifacts that often compromise in vivo preparations. While conventional resonance scanner-based microscopes can eliminate motion along the horizontal plane, Atlas offers motion correction across all 3 dimensions. Fifth, 3D photostimulation with imaging brings the ultimate level of modern optogenetics into your lab. With FEMTO3D Atlas, you can simultaneously stimulate cells that express your opsin or other photosensitive molecules and record from the same or different cell population. This webinar is structured along the main features of FEMTO3D Atlas. I will guide you through each scan mode and illustrate their value to scientific discovery by summarizing high-profile publications that use FEMTO3D Atlas. In the first part, I will show you how this product can give you an advantage in imaging at the network level at cellular scale via high-speed arbitrary frame and volume scanning, 3D random access point scanning, 3D chessboard, and multi-cube scanning. In the second part, I demonstrate how a multitude of sub-micrometer sized compartments can be imaged with FEMTO3D Atlas. In the third part, I will show you how the system can be used for simultaneous photostimulation and imaging in 3D. High-speed arbitrary frame scanning mode offers planar scanning just as any conventional microscope. Yet, already in this basic mode, FEMTO3D Atlas gives you an advantage over other microscopes by providing faster speed, up to 40 frames per second full frame. Another advantage is that the plane being scanned can be rotated to any angle in space. The left panel shows the relative position of the objective's focal plane in blue and the actual 2-photon focal plane in pink. The movement of the pink plane exemplifies the possibilities Atlas offers you to orient the field of view in a three-dimensional tissue. To the right, the actual field of view containing GCamp expressing green fluorescent cells is rotated. In summary, FEMTO3D Atlas supersedes conventional galvo and resonance systems not only in speed, but also in rotational freedom. Multiphoton microscopy became the method of choice to study brain function at the cellular level. The brain consists of billions of cells organized into local circuits arranged in 3D space. Conventional microscopes that rely on optical sectioning to cover 3D space are not well suited to capture the dynamics of neuronal circuits. In contrast, FEMTO3D Atlas is not bound to planes and can be used to record from cells scattered in 3D. This animation captures the quintessence of the difference between conventional scanning and FEMTO3D Atlas. While sampling rate in resonant scanner-based microscopes can reach 30 Hz in a single plane, Atlas captures fluorescence changes with up to kilohertz speed by scanning precisely at the location of each cell of interest. The most important capability of FEMTO3D Atlas stems from the motionless acousto-optic scanning technology powering this microscope platform. Acousto-optic scanning means that the focus can jump at any moment to any point in the volume. Thus, unlike conventional microscopes, FEMTO3D Atlas is not bound to a plane and inherently allows 3D random access scanning. On this slide, you can see an example recording performed with the 3D random access point scanning mode to sample fluorescence changes from 1778 cells in the mouse visual cortex. To the left, you can see the recorded cell locations in space. In the middle, the time series measured from all the cells on a heat map. Note the ensemble of the synchronously active cells appearing as a red line on the map. This very compact heat map view is expanded for the 200 most responsive cells in the plot to the right. The 3D random access scanning mode we've just discussed allows recording from thousands of cells. The 3D chessboard scanning mode extends the point scans to small squares around the cells. You can record from up to 300 cells that can be as far as 500 microns from each other in depth. On the video on the left, you can see the 3D chessboard scan of numerous cells scattered in a volume. Since 3D visualizations can be difficult to interpret, FEMTO3D Atlas was programmed to arrange your selected cells into an organized, straightforward visualization. In the video on the right, you can see how the activity of cells can be inspected while a mouse is looking at the visual stimulus. In 3D chessboard scanning mode, your signal quality can surpass that of full-frame scanning. With conventional microscopes, signals are captured from all cells and their surrounding neuropil, sectioned by the current focal plane. This approach is not optimal when we want to capture changes in fluorescence occurring in networks of cells because much of the scanning time is spent on places that do not contain useful signal. The 3D chessboard scanning mode allows you to focus only on the cells of interest, thereby getting better results. In this example, we selected the same 15 cells in two settings, with a conventional resonance scope and with FEMTO3D Atlas. Looking at the fluorescence time series from the two systems, the advantage of FEMTO3D Atlas chessboard scanning becomes apparent. The time saved by scanning only these cells and the traces acquired with Atlas have much higher signal-to-noise ratio. This massive increase in signal quality is especially important when activity is weak. In summary, Atlas is capable of spending scanning time only on your cells of interest, thereby increasing signal quality and sampling rate. 3D chessboard scanning is especially valuable for those performing awake in vivo activity recordings. Signal quality in awake preparations is often compromised by sample movement. Using FEMTO3D Atlas, you can correct for such motion-induced artifacts, as you can see on these two videos. To appreciate this feature, please look at the two example time series taken from this recording. The raw signals contain motion-induced fluorescence changes that can confound the interpretation of your data. On the motion-corrected time series, you can see that in the motion-compensated signal, only actual fluorescence changes remain. In other words, motion correction brings you a massive increase in signal-to-noise ratio, quantified on the bar plot to the right. The value of 3D chessboard scanning was recently showcased in a paper that demonstrated vision restoration using near-infrared stimulation. The authors of this science paper used 3D chessboard scanning to record calcium activity from multiple layers of the explant human retina. Keeping the human retina responsive to light stimulation is very challenging, but the authors could leverage the efficiency of FEMTO3D Atlas and cover cells in multiple retinal layers in one go. With a conventional microscope, these experiments would have taken much longer and would have required more of human retina samples, which are incredibly difficult to source. The fluorescence traces to the right show the responses of different retinal cell types to increasing levels of light stimulus. FEMTO3D Atlas and its 3D chessboard scanning mode was essential to obtain the results of another high-profile paper coming from the Loshonzi lab. The authors combined functional imaging by FEMTO3D Atlas with post-hoc immunohistochemistry to link morphology, function, and molecular identity of inhibitory cell types in the hippocampus. The leftmost cartoon depicts an awake mouse being scanned by FEMTO3D Atlas. Interneurons expressing GCAM6 were scanned with 3D chessboard scanning and aligned with confocal images, and you can see the distinct inhibitory cell types denoted by different colors. Thanks to the high resolution of functional imaging provided by FEMTO3D Atlas, the authors gained new insights into the collective organization of local inhibitory circuits supporting navigation and mnemonic functions of the hippocampus. The 3D chessboard scanning discussed so far can be extended into the so-called 3D multi-cube scanning by making the measured areas turn into volumes. In this mode, each cell is covered by a cubic recording area, further increasing signal quality and allowing for motion correction across all spatial dimensions. While this will have an effect on the speed of recording, this scanning type is especially advantageous if you have motion from all three directions and need information from all subcompartments. This type of recording may also come handy if you want to ensure the proper positioning of recording ROIs on the cells over a longer period of time, even for weeks or months. It is not only the network and cellular level where FEMTO3D Atlas offers advantages in imaging, but also the subcellular level when investigating dendrites and spines, for example. In the second part of the webinar, we will focus on these. The previously discussed high-speed arbitrary frame and volume scanning and 3D random access point scanning possibilities can also be used on this level as well. Furthermore, we'll explore the advantages of new modes like ribbon, snake, and multi-line scanning. High-speed arbitrary frame scanning takes us to the smaller level, since it can also be useful when recording from smaller structures. This example illustrates that thanks to the ability to tilt and rotate the frame, the apical dendrites of the neurons got aligned with the scanning plane. Thus, the spatiotemporal activity patterns of these long segments passing multiple cortical layers could be recorded without requiring lengthy and painstaking optical sectioning. 3D random access point scanning can also be used to record from subcellular processes. This movie shows a 3D recording of fluorescent signals at multiple locations along the dendritic arbor of the cell. The corresponding somatic membrane potential and local field potential signal are shown at the bottom in green and red. Spheres represent about 100 pre-selected locations scanned in 3D with a very fast, more than 300 Hz repetition rate. The color and size of the spheres vary with the changes in relative fluorescence. This example illustrates how FEMTO3D Atlas can be used to sample about 100 locations at very high speed and thus enable studying the temporal shaping of activity along the three-dimensional arborization and soma of a neuron. FEMTO3D Atlas offers a range of scanning modes and gives you the freedom to balance recording speed and spatial coverage. 3D multi-line scanning extends scanning points to lines, which can be spatially oriented. By using lines instead of points to sample the elements of interest, you won't lose recording speed. Moreover, this will allow you to retain important spatial information to counteract the effects of tissue movement. Here, we show the recording of numerous dendritic spines on a cortical neuron with a speed that allows voltage imaging. You can also observe the effect of motion correction on the curves on the right side of the slide. Ribbon scanning records free-form ribbon-like surfaces in 3D around the elements of interest. In this example, dendritic segments. Fluorescence changes both at the dendrite and at its spines can be recorded as illustrated on the right. No matter how much dendrites are winding in any direction of space, FEMTO3D Atlas can record any convoluted thin structure or several structures simultaneously. This slide demonstrates that the motion correction feature of FEMTO3D Atlas can also be applied on the subcellular level. In this example, we plotted the calcium transients of dendritic spines recorded with ribbon scanning without and with motion correction. You can appreciate the significant improvement in the signal-to-noise ratio of the reconstructed signal on the bar plot. Snake scanning is the 3D extension of ribbon scanning and can be highly useful when imaging subcellular features in behaving animals. Snake scanning can facilitate your experiment in two ways. First, scanning a volume around your thin object ensures that the fluorescence signal remains uncompromised despite motion artifacts. Second, for those who want to analyze the computational roles of dendritic spines, snake scanning offers a very efficient way to cover all the spines along a segment. Manually selecting each individual spine would be extremely laborious and error-prone, whereas in a snake scan, all the spines located around the dendrite are covered and can be analyzed. This is what you can see on the dendrites in the middle. Spines can be isolated on the projections of the snake scan, and their fluorescent traces can be exported for analysis. This way, no spine can hide behind other elements from the microscope's point of view. Here you can see an excerpt from a high-profile paper showcasing a subcellular scanning mode. The Fitzpatrick lab used the high-throughput ribbon scanning mode to map the functional organization of callosal connectivity in mice, imaging dendritic spines of single neurons in the primary visual cortex, combined with optogenetic stimulation of the presynaptic neural population in the contralateral hemisphere. They had GCAM6 expressed in one hemisphere and applied transcallosal stimulation using optogenetics. In the middle, you can see the projection of a neuron expressing GCAM6 and its dendritic segments selected for imaging during visual stimulation and optogenetic activation. The red curves show the tuning curves of individual spines to visual stimulus, and to the right, their response to callosal optogenetic activation is shown. In essence, with the help of the acousto-optical technology, they were able to perform these challenging experiments which require high spatial and temporal resolution and in vivo motion correction. I hope that our tour of Atlas features could convince you that one can hardly come up with an experimental scenario that could not be covered by the unprecedented versatility of FEMTO3D Atlas. Whether imaging frames, cells, or submicron features, anyone can find the optimal scanning mode with Atlas. The first two chapters of today's webinar were about reading out changes in fluorescence across large volumes, from up to thousands of cells with unprecedented speed. The next few slides introduce how Atlas can serve those who also want to write changes into neurons via optogenetics or uncaging. With FEMTO3D Atlas, you can simultaneously read and write activity into cells scattered in a 3D volume. In this example, you may observe the simultaneous recording and optical stimulation of 30 neurons in different cortical depths. Cells in blue squares represent activity imaging, whereas smaller red squares flash when optogenetic excitation is delivered to a cell. The neurons express both the activity sensor GCAMP and an opsin, and, as it can be seen on the traces, they readily respond to the precise stimulation. The speed of recording has of course been slowed down considerably for the purpose of this demonstration. In some cases, a very precise pattern of excitation is required. Using Atlas, you have the freedom to cover your cells or other elements with a range of sophisticated patterns or free drawings. All this in 3D. Your excitation pattern may be as diverse as when using an SLM, but with higher spatio-temporal precision. This precision can also help when you would want to ensure that the neuropil excitation is kept at minimum. Many labs have already adopted acousto-optic microscopes from Femtonics and have successfully published new results in top neuroscience journals. We believe that one can hardly imagine a better endorsement of our FEMTO3D Atlas platform than a multitude of papers from leading neuroscience laboratories published in Science, Neuron, Nature Methods, and many others. We are confident that the mature technology powering FEMTO3D Atlas in combination with our dedicated scientific and technological support team can also take your research to a new level and contribute to similar impactful results. Thanks for being here with us today for this webinar. We hope we could get you as excited as we are about the powerful features of Atlas that sets this microscopy platform apart from what conventional scopes can offer. This presentation remains accessible on the Femtonics YouTube channel, where you can also view our previous webinars discussing the acousto-optical technology itself and our conventional multiphoton microscopy platform with galvo and resonance scanning, preferable for those on a constrained budget.
Hi, everybody. Welcome back. We've received quite a few questions, so thank you for your activity. Tomasz, I think since this is an application-based webinar, most of them are to you. So let's begin. This one's about multi-line scanning. When talking about 3D multi-line scanning, you said something like, by using lines instead of points for sampling, you won't lose recording speed. How is this possible?
Well, as paradoxical as this may sound, it's very logical. Since in Atlas, we do use this genius engineerical feature, the drift scanning, which you can find, material in our former webinars, which you can check back on our website and YouTube channel. We do explain there that basically drift scan is about extending scanning points into lines, and drawing these lines takes the same time as scanning a single point. So there is some little signal to noise ratio lost, but you get a real fast end image like recording there. So that is the explanation why you don't lose any time when you record lines instead of points, keeping spatial information, keeping very valuable spatial information in imaging. Yes, thanks for the question.
Thank you. There's more on chessboard scanning, for example. Chessboard scanning seems useful in our in vivo research. How do you define the size of the tiles to maximize the number of scanned cells while still ensuring the elimination of motion artifacts?
Very good question, to the point. So yes, you're absolutely right. One of our nice special scan feature, the chessboard scanning, is right there for in vivo network-like cellular imaging in 3 dimensions. And the software of Atlas gives you a nice liberty on defining the size and resolution and edge size of those chessboard tiles so that they fit your spatial resolution and temporal resolution needs. So if you observe your tissue and it has a relatively large movement, then you will need a little bit larger chessboard tile to keep the cells inside and then allow for motion correction. Then you can again choose if you want a higher resolution within the cell and less cell to record, or it's enough to have a little bit lower resolution and you can record a bunch of cells. So again, it's a game of balance, and you have the liberty to choose the size of the chessboard dice according to your needs.
Thank you. And also regarding chessboard scanning, can you please share more on how to do chronic experiments and how do you find the same cells with the exact coordinates?
Important question. Thank you for that as well. Because a nice feature of Atlas that you can actually do recordings on a longer term as well. So when I say longer term, that means you can do it day after day, week after week, sometimes month after month. And when you do experiments like this, oftentimes the need arises to find the very same cells, the very same area. And normally what we use for that in cortical physiology, that's the vessel pattern, the vascular pattern on the surface of the cortex, which can be imaged with our camera mode or in 2-photon mode as well. And it makes it quite easy to find the same area. And then afterwards, if you reload, because that's also possible, if you reload the properties of the former recording, then you will of course record the very same area with the very same parameters day after day, week after week. We also have a little trick when we put fluorescent beads into the cover glass of the craniotomy, and then the pattern of the beads leads us to the same area. But also we can, for different kinds of tissues, we may work out different tricks which helps you doing these recordings on the long run. We are eager to give you hints and tricks. Would you want to submit your question to us? So we are here to help in these cases as well.
Okay, thank you. Off to high-speed arbitrary frame scanning. How much can you tilt the focal plane?
Well, in fact, as you have seen in the webinar, there was this nice movie in which you have seen the cortical pyramidal cells from the side. That also shows that in the case of the high-speed arbitrary frame scan with all these drift scanning properties, there is no constraint on how much you can tilt and rotate your field of view. So you can use all the many times 360 degrees of freedom what you have to turn, tilt, rotate the field of view. I have to mention though, and you have to keep in mind, that if you tilt the field of view beyond a given point, due to the fact that the 2-photon point spread function or focal point is elongated in space, that means that in Z it's a little bit longer than in X and Y, you're gonna perceive some distortion in the image, but that doesn't mean any kind of data loss. So again, you have the full liberty of tilting and rotating your focal plane with Atlas. And the software helps you to do that easily.
Thank you. Let's move on to photostimulation. How fast can you switch between imaging and photostimulation?
Yeah, photostimulation is an important topic and an important feature of Atlas. You can photostimulate just as well as you can record. And it would be important for all of us to try and be able to do simultaneous photostimulation and recording. But of course, in these systems, this is just a virtual simultaneity, which means that you switch in between stimulation and recording. And it's an important question, what is the fastest way to switch in between the two? In fact, theoretically, the theoretical limit of switching between the two in Atlas would be a single AO cycle, which in this case, and AO cycle, that means how fast you can switch in the AO crystals between two addresses. And in, again, for information, refer please back to the former earlier seminars or webinars, whatever they are. So in the Atlas system, the shortest time you can do that, that's 30 microseconds, which is very short. But of course, that's more of a theoretical limit since it applies when you image and stimulate a single point in space. They can be different points, of course, but a single point. Normally, what you want to image and stimulate, they may be frames or the fastest lines. And then in that case, the shortest time you can switch in between them is your frame rate, both in the case of lines and areas. So it's going to be frame rate, and then you can do an interleaved imaging stimulating protocol, and then afterwards, by excluding the uh, stimulated frames, you can extrapolate in between the imaged ones, gaining back your data. And then someone also asked if we can switch off, if we have to switch off the PMTs during stimulation. Normally, because of the difference of the wavelength, you would not have to do that, but for a safety reason, our systems are equipped with gated PMTs, and normally we switch them off during the time of stimulation, and that can be easily done in the protocol editor of the MES software, of the Atlas software. Yes, so that is the short answer to that question. Thank you.
Thank you. Another on photostimulation. What do you find to be the most effective photostimulation pattern for cortical cells, and how can you define them?
Very good question. Thank you. Although I wouldn't talk about the most effective pattern, since I don't think anyone ever found already the most effective one, especially as each and every preparation differs from each other. But the point is that Atlas and the software gives you the liberty of choosing practically any kind of pattern you wanted. So you can generate different patterns in 3D, which can be either just a single point, axes, areas, spirals, whatever suits you. And you can even individually cover the cells or the cellular element you want it particularly to stimulate, or where you want to do, for example, uncaging. So you have a full liberty of choosing your patterns in 3D, even more than you would do with an SLM. And we have also talked about that in one of the earlier webinars.
Thank you. This one's on external triggers. Could you maybe describe some possibilities for setting up external triggers or ways to trigger image acquisition with external devices?
Yes, important question. Atlas is equipped with a full input-output system where you have both analog and digital inputs and outputs, and they can be handled easily from the software. So you can trigger both the imaging from the outside, but you can also trigger from the microscope. And you can also not just trigger, but send out like time flags to all different kinds of equipment, be it other imaging equipment, electrophysiology, behavioral setups. You can use those input-output sets to get input from the behavioral setup and start imaging at a given point, or just get a time flag from somewhere from the behavior. But you can also do the opposite. You can send out trigger signals, when to start electrophysiology, etc., etc. Basically, the possibilities here in this case are unlimited. The bandwidth of the input-output device is really wide.
Thank you. And as I see it, this question concludes our Q&A session. And I really appreciate the activity that I saw from our audience today. And thank you for being here with us today. I hope you found the capabilities of FEMTO3D Atlas exciting. And do stay tuned for our upcoming webinars where we broadcast a live in vivo imaging session with Atlas and discuss our software in more detail and introduce our partner labs who are using Atlas in their research on a day-to-day basis. And we also hope to see you in January at the online version of the SFM meeting called the Global Connectome.
Also looking forward to meet you in person maybe later in case of trainings and
personal demonstrations.
Demonstrations, yes.
Okay, yes, absolutely.
And I also appreciate the activity. Thank you.
Thank you. Stay safe and healthy and hope to see you soon. Bye-bye.
See you later. Bye.